Modern biosecurity operates on a highly specific assumption. Biological threats rely on engineered pathogens or permanent alterations to a target's DNA. Traditional genetic engineering, using tools like CRISPR, cuts into the target's genome. This creates a permanent genetic scar, and because the modification is written into the DNA, the body must undergo cell division to replicate the change. That cellular trail is forensically trackable. A new category of synthetic biology circumvents genomic defense frameworks by targeting the intermediate messenger, RNA. This cellular model demonstrates the mechanical difference. On the left, a DNA edit locks a persistent mutation into the cell. On the right, an RNA edit issues a temporary instruction, produces a target protein, and then dissolves. The underlying DNA remains untouched. Defense frameworks designed to sequence permanent biological pathogens are unequipped for these interventions. Tracking these threats requires monitoring transient biochemical events that leave no permanent genomic trace. The foundation of this threat model is programmable RNA editing, a biological mechanic directly inspired by cephalopods. Scaling that mechanic from a laboratory curiosity into a population-level event requires a distribution mechanism. For widespread dissemination, researchers are investigating aerosolized viral vectors. The final required component is operational timing. The edit remains dormant until a real-time environmental trigger, like a specific chemical, forces the activation. The convergence of cephalopod-inspired RNA editors, viral vector delivery, and environmental triggers establishes the technical requirements for a civilian-scale biological intervention. This technological synthesis allows for a real-time biological attack that actively rewrites the target's protein expression, then effectively deletes its own evidence. Octopuses routinely survive extreme environmental stress, like sharp temperature shifts, by adapting their biology immediately rather than waiting for generational evolution. They achieve this through a family of proteins known as ADAR, adenosine deaminase acting on RNA. ADAR proteins operate as biological proofreaders. They intercept messenger RNA strands inside the cell, swapping specific nucleotides to rewrite the genetic code. This produces custom designer proteins, allowing the organism to survive immediate environmental stress while entirely bypassing its DNA blueprint. Synthetic biology programs such as LEAPR or Restore are actively mimicking this ADAR process to engineer programmable RNA editors for human applications. The therapeutic goal of these editors is to trigger rapid temporary tissue repair or fix mutations without risking a permanent alteration to a patient's genome. Mastering the cephalopod recoding mechanism provides the capability to rewrite a host's physical state on demand. Traditional genetic manipulation has an operational limitation. For a DNA edit to alter a host's phenotype, the altered cells must divide and replicate over days or weeks to propagate the change throughout the tissue. RNA editing bypasses replication. It operates directly on active protein synthesis. Because RNA is constantly being translated by ribosomes, the programmed editor takes effect the moment it enters the cell. This bypass results in near-instantaneous physiological shifts within the host. In clinical settings, this immediacy allows researchers to counteract a fast-acting toxin or close a wound in real time, dictating the body's response during a crisis. In a weaponized context, this transformations what would normally be a slow-incubating biological pathogen into an immediate tactical capability. Scaling cellular recoding to a population requires a systemic distribution strategy. To dose a civilian population with synthetic RNA editors, you need an efficient delivery vehicle capable of penetrating human cell walls across millions of targets. The primary delivery tools developed for gene therapy are viral vectors, specifically adeno-associated virus or lentivirus. These vectors contain a technical vulnerability. The viral vehicle is strictly a transport mechanism, agnostic to its payload. In a swap scenario, a vector designed for healing is hijacked. Therapeutic instructions are removed and replaced with different program sequences. A weaponized payload targets different pathways, such as those governing neurological signaling or metabolic function, causing rapid cellular disruption. Because the recoding mechanic bypasses cell division, this altered virus begins hijacking civilian RNA the moment it penetrates the cellular membrane. The delivery systems currently being optimized for public health infrastructure share the same engineering path required to deploy a bioweapon. The temporary nature of RNA-based interventions presents a distinct biosecurity challenge. RNA is a fragile molecule. It naturally degrades and is flushed out of the cellular system within hours of completing protein synthesis. This chart visualizes the washout effect. The solid line shows physical damage peaking. The dashed line tracks the RNA blueprint, which spikes briefly, then degrades completely to zero. This creates a forensic gap. The attack's biological blueprint dissolves. Traditional biosecurity relies on isolating an engineered pathogen and sequencing its DNA to identify its origin. The washout effect makes an engineered attack difficult to distinguish from a novel, naturally occurring illness. Operationalizing this threat requires control over mass dissemination and activation timing. This comparison table outlines the functional difference. Therapeutic tools are activated by localized signals, like cellular injury. Weaponized payloads are engineered for systemic, environmental triggers. The most advanced application involves binary triggers. An engineered RNA editor remains inert within a host and synthesizes no proteins until a secondary input, like a specific chemical, activates the sequence. This allows an adversary to pre-position a biological payload across a population and trigger the response simultaneously. While clinical gene therapy relies on localized injections, research is accelerating on nebulized or aerosolized viral delivery for population-scale distribution. Combining mass aerosolization with dormant binary triggers provides a system for rapid, widespread physiological disruption across a population center. For defense auditors modeling the 2026 biological threat landscape, theoretical mechanics must be translated into observable developments. The dividing line between a medical therapy and a biochemical weapon is found in payload stability, the delivery method, and the trigger mechanism. One indicator is gain-of-function research actively focused on making transient RNA editors more stable or more potent. Increased potency in a medical tool translates directly into increased efficacy in a weaponized payload. A second indicator is the funding architecture of gene delivery, specifically platforms transitioning away from intravenous application toward aerosolized or nebulized formats. Auditors also track clinical studies testing synthetic RNA editors engineered to remain inert until a secondary chemical activation is introduced. Monitoring these specific development vectors is necessary to address the blind spots of current DNA-focused defense systems. These technological leaps will not be limited to military black sites. They require vast resources and are often openly funded as academic and public health research. The discrete biological components required to construct these payloads are currently being mapped and optimized under the umbrella of medical breakthroughs. Defense auditors reviewing government contracts must focus on the intersection of public health and operational security. Procurement requests for large-scale aerosolized RNA delivery platforms warrant scrutiny. If a project is listed under pandemic response but mandates classified security clearances, the dual-use threshold has likely been crossed. When a public health mechanism requires classified operational security to develop, it indicates that the technology has moved from academic inquiry toward tactical application.